Hello, everyone, and welcome to today's webinar, Multi-omics Approach to Understanding PD-L1 Resistance in Cancer. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more about our sponsor, please visit www.sonybiotechnology.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education's Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Moshe Sade Feldman, the instructor in medicine and the director of the Translational Cancer Immunology Lab at the Massachusetts General Hospital Cancer Center, and Dr. Russell Jenkins, assistant professor and member of the Faculty of Medicine at the Massachusetts General Hospital Cancer Center. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Feldman and Dr. Jenkins will now begin their presentations.
Thank you, Michelle, for this kind introduction. Here are disclosures. So I'll first start with a quick background on checkpoint blockade therapies. I'll start with the how and why, so how those type of therapies actually work. So checkpoint therapies actually take advantage of the natural process of T-cell activation. Whenever a T-cell gets activated in response to stimulation, could be cancer, could be infection, there's an upregulation of inhibitory receptors on the cell surface of those cells. And this actually controls the amount of activation of the cells, preventing them from over-activating and eventually phenomena that can lead to tissue damage and autoimmune-like disorders. And the way that checkpoint therapies work, they actually block the co-inhibitory receptors, and by doing so, they're unleashing pre-existing immune responses of the T-cells against the tumor cells. One thing we need to know about checkpoint therapy, that these therapies actually depend on the ability of cytotoxic CD8 T-cells to recognize neoantigens that are expressed on the cell surface of the tumor cells in the context of class 1 presentation. So what's so unique about checkpoint therapy? So checkpoint therapy have succeeded where conventional therapies have failed, and if we, in the terms of inducing more durable responses in a wide variety of metastatic disease, a fact that eventually led to the rapid approval of these drugs in a wide variety of cancers in recent years, with melanoma being the first one in 2011. However, although even if we take melanoma that has one of the highest mutation rates, still most of the patients do not respond to those therapies, and those who do respond eventually relapse within the first year and a half. And the main question that we ask ourselves, what actually drives tumor to escape from checkpoint therapy? Why some patients respond and why some patients do not respond to therapy? And in recent years, there's been extensive research trying to answer those questions. And when we look what are actually the drivers or mechanisms or factors that are associated with either response or lack of response, so we can actually break it into two. So the upper part talks about what we may call the acquired resistance, patients that have an initial response to therapy, but after a while they relapse and the tumor progresses. And patients, if we look at the lower part, these are the primary resistance tumors. These are tumors that do not respond to these types of therapy. This could be due to lack of infiltrating or targeting T-cells within the tumors, or defects that are already present within the tumors prior to therapy, such as loss of antigen presentation capabilities. If we look at the upper part, So what actually drives the response? So we know that tumor mutational burden, the more mutations the tumor have, the more targets they have for the immune system to recognize. We need a functional immune system in those tumors, most likely dendritic cells that can present those antigens to the tumor, to the T-cells. We need the T-cells that have a cytotoxic functionality that can actually recognize those tumors. But then when we have these kind of responses, these responses, as I mentioned, are usually limited in time, and eventually there's a relapse in most of the cases. And again, this could be due to acquired mutations that the tumor have acquired that enable him to evade immune destruction or the detection of the tumor by the immune system, or by generating or altering the immune system by generating a more tumor-supporting microenvironment, such as the upregulation of an additional co-inhibitor receptors or the attraction of suppressive cells that can actually suppress the activity of the cytotoxic CD8 T-cells. However, if we look at those studies, there are those studies that have been done in recent years have major limitations. First of all, they have a low predictive power. For instance, if we take tumor mutation burden as a standard that has been evaluated in many types of cancers. So if we compare how good is TMB in our ability to separate between patients that are more likely to respond, we see that there is a big overlap between patients who respond and have a low tumor mutation burden versus patients who do not respond and have a high tumor mutation burden. And we see that there's a big overlap, and in many studies you cannot separate between the responders or patients that will benefit in terms of survival by using those specific markers. The other limitation is the usage of predefined immune markers that actually limits our ability to identify new factors that can actually elicit an immune response or associated with either success or failures of those therapies. The usage of bulk RNA-seq data that pretty much limits our ability to infer specific cellular states or markers in specific cells or the contribution of specific cells, as we are looking at an average of expression within a given tumor and not a specific expression within specific cells. And again, I think the other limitation that was that most of the studies that were using unbiased approaches before in recent years actually focused mostly on the tumor cells, but pretty much ignored the microenvironment or the immune system, which is also crucial for the success of these types of therapies. So what's the approach that we decided to take? So as the title mentioned, we do use a multi-omics unbiased approach to actually answer the question. We don't start with a specific hypothesis. We start with a problem that we have, and in this case is melanoma patients that are treated with checkpoint therapy. We have the patients that respond, patients who do not respond, and now we want to find why some respond to therapy while others do not respond. So actually the way the approach that we take is being broken into two steps. The first step is the discovery phase, when we use really both genomic and transcriptomic unbiased approaches to discover the landscape of immune cells within those tumors. Once we identified and classified those immune states and cells, we identify which one of them is actually associated with a clinical outcome. The second part is the validation phase. So once we identify either a cell or a specific gene that is associated with a clinical outcome, then we start to build our hypotheses, and then we experimentally check them. This could be done by using ex vivo system, in vivo system, and other functional assays. So as I mentioned, some of the patients that are treated with checkpoint therapy respond, but the majority of patients do not respond, and our major question was why some patients respond to therapy and why some patients do not. And we decided to focus on melanoma, and the approach that we applied for that was using single-cell RNA sequencing. So the cohort in general that we analyzed was composed of 32 patients, and as you can see here in the upper right part, it actually breaks the cohort distribution. We had 31 non-responding lesions that were taken from patients that were treated with checkpoint therapy. We had 17 responding lesions that were taken at baseline or on treatment, and we also had 11 patients that had matched pre and on therapy samples. The way that we did this type of study, we got fresh biopsies from the surgery operation room. We rapidly dissociated the samples. We isolated the immune cells using a cell sorter, and then performed the single-cell RNA sequencing using the SMART-seq2 protocol that is a full-length protocol that has the best coverage in terms of single-cell methods today. We also incorporate this with other methods, looking at the genomic level to see whether there's mutations that can also interfere with the immune-tumor interaction, such as loss of class 1 presentation capabilities and the interferon gamma pathways. And eventually, we performed an integrated analysis to incorporate all those parameters, perform validation experiments, and associate everything with the clinical outcomes. Two things that I would like to emphasize or move on is that why do we needed to sort the cells, and why do single-cell approaches are better than the bulk approaches? So single-cell enables us actually, as I mentioned earlier, to look at specific transcripts within individual cells, and to look at specific pathways within individual cells, to look at the compositions of those tumors and how those cells relate to clinical outcomes. And again, unlike single-cell RNA sequencing, bulk RNA sequencing gives us only the average of what's happening in the tumor, and we cannot infer the states or specific genes within individual cells. And the example that I show here below actually shows that there's been several research talking about specific genes that are associated with clinical outcome, but then when we look at those genes and their association, for instance, with CD3 that reflects the T-cell infiltration within those tumors, we see that there's a very high correlation between those genes, that actually the genes that we see by using bulk RNA-seq are usually correlated with the amount of or the degree of infiltration of a specific immune cells, and in melanoma, it's mostly T-cells. So in that correction, when we look at or use bulk RNA-seq data, and that's, I think, one of the main limitations, you will see transcripts that belong to a specific cell if this cell is highly abundant within the tumor. Limitation for single cell. So single cell is a very good method, but it has a limitation that we need to recognize. It's more noisier than the bulk RNA-seq data, and that's due to technical limitations and biological factors. There's more heterogeneity. Also, single cell, unlike bulk RNA-seq, has a higher dropout rate and a lower capture efficiency. So not all the time we will capture all transcripts within a given cell. The second part, which is to the right, why do we actually need to sort? And this is very important because not the quality of the sample is not even among all patients. Sometimes the sample is very necrotic, and the amount of viable cells within a given tumor is very, very low, as you can see here in the upper left corner. So when we start the sort, we actually have the ability, even if we are giving a tumor with very low viability, we can actually focus on the good viable cells, and by using or excluding live dead markers. Another thing that we incorporate into our cell sorting is the usage of the CD235A marker. And the reason that we did that, we noticed that, especially it's relevant when we look at the tumor side, that there's a lot of erythrocytes progenitors that are within the tumor, and these are cells that are usually CD45 negative, and the lower plots actually shows you that what happens in a tumor that you have a lot of those cells. So you will see those cells as CD45 minus, but then when you look at their class 1 expression, which is one of the hallmark markers of red blood cells and red blood cells progenitors, they do not have any class 1 molecules. So that actually helps us to distinguish between those populations of cells that eventually their RNA content is minimal, and eventually the quality that we get from the sequencing is very bad if we integrate those into the tumors. They can also falsify or give us negative results in terms of that we might think that we're getting on the CD45 negative cells, which is mostly the tumor compartment, and then we might think, okay, these tumors are lacking class 1 expression, but they're actually not, because we are getting on this very dominant population of erythrocyte progenitors. So this is one additional thing that really help us to distinguish the good and the bad cells, and actually making sure that we are getting on the right populations. So as I mentioned in the two previous slides before, we decided to focus our efforts on the melanoma cohort of patients treated with checkpoint therapy. We initially used the single-cell RNA approach in an unbiased way to define the immune landscape in melanoma patients. So by doing this type of analysis, we ended up with 11 clusters by using an unbiased k-mean clustering approach. And we ended up with 2 B-cell clusters, 2 myeloid clusters, and the remaining clusters, the 7 remaining clusters, were mostly composed of lymphocytes at different cellular states. And the next question that we want to ask ourselves, which one of those cell types and states are actually associated with clinical outcome? And when performing this type of analysis, we saw that there were actually 6 clusters associated either with bad or good clinical outcome. We saw that the B-cell cluster was associated with, it was more enriched in the responder compartment. The 2 myeloid cluster were more significantly enriched in the non-responders, and 3 clusters that were mostly composed of CD8 T-cells. One of them reflecting a memory phenotype was mostly enriched in the responders, and 2 clusters that were mostly associated with T-cell exhaustion and dysfunctional phenotypes were enriched in the non-responders tumors. Now, since the majority of cells identified within our cohorts were mostly CD8 T-cells, and due to the fact that we know that CD8 T-cells are crucial for the success of checkpoint therapies, and that 3 of the clusters that were either associated with response or lack of response were mostly enriched for CD8 T-cells, we decided to focus the rest of our analysis on this cell compartment. So we started by focusing on the CD8 T-cell compartment, and we started a very unbiased analysis by breaking the CD8 T-cells into 2 clusters, and we ended up with what we call the CD8B, the bad cluster, that's mostly enriched with exhaustion markers and markers that are usually linked to dysfunctionality of the cells. And the second cluster was the CD8G cluster, the blue cluster, that was mostly enriched for self-renewal genes and genes that are mostly associated with stemness and memory of CD8 T-cells. And the first thing that we want to ask ourselves, how are those 2 cellular states associated with the clinical outcomes? So when we looked at the responders, we see that there's a significant enrichment for the CD8 good, the good cluster, the cluster that's associated with memory cells. And when we look at non-responders, we see that there's a significant enrichment for the bad cluster. Interestingly, when we look at patients, we don't see that the responders have only the bad cells or the CD8B, the ones that are dysfunctional, and the responders have cells that are only the good cluster, the memory cells. We see that there's a coexistence of those 2 populations. However, we see that in most cases, in non-responders, we see an enrichment for the dysfunctional cells, and in responders, we see an enrichment for the good cells, the memory-like cells. And the upper plots actually show you a patient who initially responded to therapy and after a year developed resistance. And at the time of resistance, we see that there's actually a switch within those 2 cellular states. Now, since we saw that those 2 cellular states coexist together in patients that are treated with checkpoint therapy, we decided to calculate the ratio between the CD8 good, the memory cluster, versus the CD8B, the dysfunctional cluster. And when looking at all samples or at baseline samples prior to therapy or on treatment, we usually see that in most cases, patients who do respond to therapy have a higher proportion of those CD8G good cluster cells versus the CD8B, the dysfunctional T cells. When we take into account the whole spectrum of ratios, we usually see that a ratio above 1, meaning that there's more of the good clusters, is a very predictive marker, a very good predictive marker of the clinical outcome. In this case, is the clinical response to checkpoint therapy in melanoma patients. Third thing that we wanted to do after we identified cell types and link them to clinical outcome, we want to see whether we can actually identify predictive single markers that are linked to response. And we looked at all cells, and within CD8 T cells, we ended up with a list of markers that were either enriched in responders or single genes that were actually associated with lack of response. And then we ask ourselves, can we actually take one of those genes or that we identified and predict the clinical outcome in an independent cohort using a method that's more applicable to the clinic? So we decided to focus on TCF7 because this gene was one of the top genes that was associated with response when looking at all cells, or we're looking specifically within the CD8 T-cells. It was also one of the most highly differential expressed genes within the CD8 G cluster, which is the memory-like cluster. So what is TCF7 or transcription factor 7? It's part of the Wnt signaling pathway. In the context of the immune system, this gene has been shown to be crucial for the self-renewal and differentiation of hematopoietic stem cell. It has also been shown in recent years to be crucial for the reinvigoration of CD8 T-cells in response to checkpoint therapy in the context of mouse chronic CMV mouse models. So the approach that we took was using a pathology-like approach, using a immunofluorescence staining on FFPE sections. So again, we collected an independent cohort of patients. You have the break here, a total of 43 samples. We had 23 responders, 23 non-responders, 20 responders. And the idea was to stain for TCF7 within CD8 T-cells because this is where we saw the gene to be associated with response. We didn't see TCF7 in B cells. We didn't see TCF7 to be significantly enriched in myeloid cells. We saw this gene to be significantly associated with response when it's expressed in CD8 T cells. So we took this approach. We stained the section. We developed a pipeline using the cell profiler algorithm that actually enables us to quantify the number of CD8 T cells that are either positive or negative for TCF7 in an unbiased way. And when we perform this type of analysis, similar to what we did when looking at the single-cell RNA-seq data, we saw that CD8 T-cells that are positive for TCF7 are significantly enriched in responders, and tumors that actually do not respond to therapy are more enriched for CD8 T-cells that are lacking TCF7 expression. Similar to the single-cell RNA-seq data, we see again that these two populations of the TCF positive and negative CD8 cells coexist in responders and non-responders. Within the responders, we see usually a higher proportion of those positive cells for TCF7. In the non-responders, we see usually a negative. Again, similar to the single cell, we calculated the ratios those two populations coexisted. We looked at all samples, baseline samples, on-treatment samples, and again, we see that we can actually predict the clinical outcome by looking at those two parameters. In addition to the clinical outcome, we see that in most patients that have a higher proportion of those cells, they also have a better outcomes in terms of the clinical survival. The next thing that we want to do once we perform this type of analysis is to identify the identity and functionality of those specific cells and cellular states. So I'll speed up a little bit the talk here. So we took the CD8 T-cell compartment. As I mentioned, we started with a very simple analysis by breaking them into two clusters. But when we dived in a little better and performed additional analysis, eventually the CD8G and the CD8B broke into three clusters each. So we have three different clusters of T-cell exhaustion or dysfunctionality. And we ended up with the CD8G with three clusters that represents three different cellular states of T-cell activation and memory. What this type of analysis actually enabled us to do was to identify specific cell surface markers that can actually enable us to pull out those specific cells from either human or mouse tumors and test their functionality. So when we perform this type of analysis, or when we pull those cells out, again, using cell sorting approaches, we compare the isolated cells that we identified by the cell surface markers to the original single cell RNA-seq data, and this is the heat map in the middle. So the red and blue to the left actually represents the single cell original data, and the plots to the right actually represents the sorted cells that we pulled out based on the cell surface markers, and resequence those to make sure that we're actually recapitulating the same cellular states that were associated either with response or lack of response. So then we performed additional types of analysis, and we asked ourselves which of those two cell states are important for the tumor eradication in response to checkpoint therapy. In this case, it was PD-1 therapy. So we pulled out those cells and we performed a series of analysis, and Russ will elaborate more about the microfluidic system, the approach that we use in order to see which one of those cellular states is more important for tumor eradication. We also performed some in vivo experiments. So the nice thing about the single-cell RNA-seq data, it doesn't also let us recognize cells that are either associated with response or lack of response, and actually let us know what are the markers that are highly expressed in non-responding tumors, and then we can find new combinations to therapy. And this is what we actually did with the in vivo experiments. Based on the human data, we identify a set of targets that are expressed on the cell surface, so it's easy to target them. And then we saw which combinations are most significantly enriched in non-responding tumors, and then we verified that we see the same things in mouse model tumors, and then we performed a dual targeting, and we saw that by dual targeting those targets that were identified in an unbiased manner in the human data can actually enable us to elect better clinical outcomes in vivo mouse models in terms of the response of the tumors and also in the survival of the mouse.
Thank you, Moshe. So to talk a little bit more about some of the functional validation studies we did, I'll give a little bit of background on some of the work that we do in our group, which essentially is creating a window into the tumor immune microenvironment. And that's the murine-derived or patient-derived organotypic tumor spheroid model, or MDOTS or PDOTS for short. So just by way of a little bit of background, we get fresh, never frozen mouse tumors or patient tumors. They're subjected to physical and enzymatic dissociation and then passed over filters to get different size multicellular tumor spheroids. And we've lots of experience and data showing that the immune contexture of these different size fractions, not very creatively named S1, S2, and S3, essentially greater than 100 micron, 40 to 100 micron, or less than 40 micron, don't differ significantly from the baseline tumor. And because they're optimally sized, we take the S2 fraction, which measures 40 to 100 micron based by size selection, and we inject that into a microfluidic chamber so that they can be cultured in a collagen hydrogel for short-term ex vivo profiling. And we can read out a variety of things on the back end of this experiment, including whether there's immune-mediated killing, production of cytokines, or transcript changes. For this talk, we'll discuss mostly the ex vivo killing. And I'll walk you through a little bit of the workflow here. So we start off with the tumor, it gets minced, and we end up with a mixture of these different multicellular tumor spheroids. And then, as I mentioned before, we end up with these different size spheroid fractions. We take the specific fraction ranging in size from 40 to 100 microns. It gets embedded in a collagen hydrogel. That gets injected into the microfluidic device, and you have a picture of what that looks like under the light microscope at day 0. And then it gets treated, usually with an anti-PD-1 antibody or some other combination treatment, and then we read out those different things on the back end. So very quickly, I wanted to just show a video, a cartoon of what this looks like in the microfluidic device. So we can see here in this animation a standard microfluidic device with the center gel region flanked by anti-parallel media channels. So the spheroids, the cells, the tumor cells and the immune cells are in these multicellular tumor spheroids in this center gel region suspended in just a couple microliters of collagen. After they've incubated for about 30 minutes at 37 degrees, we hydrate these devices with media containing either an antibody isotype control, like an IgG antibody, an anti-PD-1 antibody, or for combination treatments, you can add a small molecule or targeted therapy, and then we can observe the response of the tumor immune microenvironment over time in a given microfluidic device. is what it looks like under the microscope. You start off with spheroids at day 0, and you can see that they grow very nicely and robustly over time. This is the MC38, very immune-responsive syngeneic model. We can, again, sample the conditioned media and look for secreted cytokine changes. We can look at the immune contexture by immune fluorescence as well. So again, just to provide the background for how we did the studies with Moshe, I'll talk a little bit more about some of our proof of principle studies where we took this immune-competent PD-1 responsive MC38 syngeneic model, which has very nice in vivo response to single-agent PD-1 blockade shown on the far left, and we actually challenged it outside of the mouse with a PD-1 antibody. And this is our workflow to do that, where we get the tumor, we make our M-dots, and then we load parallel microfluidic devices and treat them either with an isotype IgG control or a PD-1 neutralizing antibody. And we have an ex vivo culture for 3 to 6 days and then perform a simple live dead assay with acridine orange propydium iodide. So this is what it looks like when the spheroids, not the mice, are treated with the PD-1 neutralizing antibody in a sensitive syngeneic model. You can see accumulation of the propydium iodide red signal and a loss of the acridine orange live signal. And we can zoom in on an individual spheroid and capture that a little bit better. And shown below is the very PD-1 resistant B16F10 model. So harkening back to what Moshe spoke about not too long ago, how do we use this sort of system for functional testing? And we had a system and the wonderful data that Moshe described where we had these different populations of CD8 T-cells that we think are responsible for driving response or non-response to PD-1 blockade. So we discussed a way that we could do this using a functional assay in the microfluidic chip. So in designing this validation study, we wanted to pick a model system that was partially responsive, but not fully responsive. And we needed to be able to characterize that we had both the CD8 good guys and the CD8 bad guys. Again, the effector memory type versus the severely dysfunctional exhausted type, sort them out, and then be able to enrich in the microfluidic device, either with pure populations or mixed populations. So for our first proof of principle study, we simply characterized that within a partially sensitive mouse model, here was the CT26 expressing a GFP marker model at 14 days, that we have a nice mix of the CD39 TIM3 double positive up in the upper right quadrant here, so double positive or DP. These are the CD8 bad guys. And then in the lower left quadrant, we have the double negative, CD39 negative, TIM3 negative, CD8 good guys. We have a nice mixture of these different populations. Shown on the right by immune fluorescence is what these tumor spheroids look like under native conditions on the middle panel, where we see our GFP staining tumor cells versus our CD8 cells in red, versus condition where we essentially spiked into the device additional CD8 T-cells in an unselected fashion. So for the workflow for the experiment to test whether the CD8 good guys or bad guys are modifying that response to PD-1 blockade, we set up the following experimental design, where tumors are grown in mice, we take them out, we generate M-dots from them, but at the same time we take the unused proportions, the S1 and S3 fraction from the original tumor prep, and then we do cell sorting based on the expression of CD3, CD8 to make sure that we've got our T-cells, and then to look by expression of those exhaustion markers, CD39, TIM3, to get either a double negative population, a double positive population, and we preserve some of the extras so that we can mix them in a one-to-one fashion. And then they're treated into the microfluidic device either with an isotype control IgG antibody or a PD-1 neutralizing antibody, cultured for 5 days, and then we assess immune-mediated killing on day 5. So I'll walk you through the bottom panel here, which essentially tells the tale. So on the far left of this graph, entitled CT26 GFP M-dots, we see the conditions with a PD-1 neutralizing antibody on the far left, where we get modest immune-mediated killing with PD-1 blockade, and this is consistent with this particular model system. If we enrich 100-fold the number of CD8 T-cells, but they're mixed in an equal proportion, the good guys and the bad guys, we don't move the needle at all. Essentially, it's as if we have added no additional CD8 T-cells. If instead we add the double negative or CD8 good guy T-cells, all of a sudden we are able to really improve upon the immune-mediated killing in response to PD-1 blockade. Importantly, these cells are not actually killing the tumor cells in the absence of PD-1 blockade. So simply enriching for CD8 T-cells on its own is not doing anything to the tumor. However, and interestingly, if we add the double positive, the CD39 positive, TIM3 positive CD8 T-cells, the bad guys, not only do we not enhance killing, but we actually blunt the killing that was observed in the unperturbed native conditions. This did point to and actually, I think, confirm the suspicions that the balance between these two CD8 T-cell states was really important for driving the immune-mediated killing.
So before we move or end this presentation, conclusion part, I do want to say a few words of PDX models and single-cell RNA sequencing that actually thing that accelerated in recent years. So PDX models, those who do not know, these are patient-derived xenografts. These are tumors that are extracted from patients. They're injected to immune-deficient mice. This tumor growth, usually people test those PDXs in the context of target therapy. And then when people start to characterize those tumors, how the drug affect those tumor cells, what people usually do, they just extract the PDX tumors. They either use the plate-based method, bulk RNA-seq data, or microfluidic systems like the 10x genomic platforms, and then sequence the tumors. And then you get those nice t-SNP plots, you get the distribution of cells and states, and sometimes, or in most cases, people have the wrong conclusions. And the reason for that is that those PDXs, when we look at their compositions within those immune deficient mice, so we think, okay, there's no immune system, so all the cells that we have, there are actually tumor cells, so we are looking at this. And in most cases, this is a wrong assumption. And the reason that I'm saying that, because we had this issue. We had a very large PDX study when we did those type of analysis, and then we started seeing strange things that are not related, we know that are not related to the tumor cells. And then when we started looking what could be the reason, we started phenotypically characterizing those PDX tumors. And if you look at the lower plots, the far left one actually shows you a PDX model that the composition of CD45 negative cells is less than 30%. And in a good tumor, we have a fraction of 70%, meaning that we have a 30% contamination. In addition to that, when we look at the human markers of those tumors, in this case, these are epithelial tumors, we see that we can enrich for those specific cells if we want to, and especially that a thing that we actually need to do. And one thing that I want to emphasize about those PDX models or combining PDX models with single-cell RNA sequencing or bulk RNA sequencing in response to treatment is the first step is actually to exclude the mouse host immune cells by performing these types of gating strategies, because again, the purity is usually low, it's usually around 40 to 50%. And on top of excluding these immune host immune cells and other stromal cells is actually the usage of markers, of human markers that we know that are expressed on those tumors, and then we pretty much make sure that the cells that we extract for the sequencing are actually the tumor cells that we targeted in vivo using different drugs. And that actually, I think, improves the data and the interpretation and the things that we see using those PDX mouse models. To conclude or to summarize what we've both Russ and me have talked about today, so the usage of multi-omics approaches or including unbiased approaches is to really understand what's happening in the setting of human malignancies, and especially in the context of checkpoint therapies. So as I mentioned earlier, these studies are usually broken into two steps. We have this discovery step that we actually enter to the area in an unbiased manner. We don't have any hypotheses. We collect the right cohort of patients to answer those questions. We perform those unbiased analyses. We identify cellular states, single genes that are associated with response or lack of response. Based on those discoveries, we start building our hypotheses, and then we check them either using a new and innovative 3D microfluidic system that can actually recapitulate the in vivo microenvironments, or using in vivo mouse models and other experimental approaches to answer those questions. Finally, I think both of us, we'd like to actually acknowledge a lot of people that help us do those types of analysis, the clinicians, of course, at Mass General and MD Anderson, people in the labs that we've been working, other collaborators that their names are presented here at the end. And thank you for listening, and now we'll be more than happy to answer any questions that you have.
Thank you, Dr. Feldman and Dr. Jenkins, for your informative presentations. We will now be pushing out a survey to the audience that will pop up on your screen. We will give you a moment to answer this. We appreciate your participation. Okay, we will now move on and start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, if most mutations in neoplastic cells affect intracellular replicative machinery of the cell, while immune system recognize only membrane abnormal antigens, how does the mutation burden correlate with immunogenicity of cancer cells?
That's a very interesting question that I would like to answer. So regardless of what pathway is being changed due to specific mutations or mutations, especially when we talk about melanoma that has one of the highest mutation rates. So every mutation that occurs within a given cell, and we have that leads to a mutated protein or product, it's being degraded through the immunoproteasome, and eventually it's being presented through class 1 molecules on the cell surface of the cells. And within those mutated proteins or peptides that are eventually being presented on the cell surface, the more mutation a given cell will have, the higher probability that this peptide or neoantigen that was specifically mutated in the cancer cell will be recognized by an immune cell. So that has nothing to do with actually what pathway is being changed or what protein is being changed inside a given cell. It could be things that, again, as mentioned, related to cell proliferation, growth, and other factors that can enhance the motility of cells. It has literally to do with how many antigens are eventually presented, and it has nothing to do with what type of protein is being mutated, because all those proteins are eventually being presented on the class 1 molecule on the cell surface. So again, the more mutations, the higher probability that this mutation will eventually be recognized by an immune cell is higher. So the higher the TMB, the higher the probability that there will be an immune recognition and then eventually a better prediction or for clinical outcomes.
Great, thank you. All right, next question. How do you know that the mouse is an appropriate model to test the drugs?
Very, very good question. So this is, I think, a problem that a lot of people encounter if they think they have a new, exciting, interesting target, and they're interested in finding out, in particular, if there's an interaction with the immune system. I will say that a lot of the novel immunotherapies started out in mouse model systems, so I think it's a very reasonable starting point if there's a novel checkpoint or another immune modulator to start off in a murine model system. Exactly which model system gets a little more complicated. When Moshe and I were working together, we picked kind of a partially sensitive model system with the hypothesis that the different CD8 T-cell states would both be represented. And we had no data. We just had to kind of pick a model system and try. And so I think that's a reasonable place to start. Now, some of the model systems are more immunogenic, more responsive. Some are less immunogenic, less responsive. I think one just has to kind of pore over the literature, figure out what other people are doing, see if the target they're interested in exploring is one that might be more active in one model system versus another, and then just do the experiment. I think one of the advantages of doing ex vivo profiling is if one has a hypothesis that a particular drug or target is going to be potentially more active in a model tumor immune microenvironment, that you can take a handful of tumors, tumor explants, and actually do the experiment ex vivo in the microfluidic culture system, as opposed to having to go straight to in vivo where you don't necessarily know exactly how to start, what to start, or if there's going to be single agent or combination efficacy. But I'd say mouse systems are still informative. They do teach us things. The fact that in Moshe's data, which was all human patient samples, they identified these discrete cell states, these discrete CD8 T-cell states, and those same states were also recovered in a mouse system, and several other groups have subsequently published similar findings in murine systems. Does point that there will be some conservation across species, but ultimately one just needs to test the hypothesis, I think.
Great, thank you. All right, next question. How do you know that the SC analysis is superior? I would expect the harsh separation of cells would in itself cause changes that are not natural but stress response.
So first of all, I have to say that I do agree with this. So if we take a given tissue that's composed of different cells, and the cells interact with each other, and when we start isolating specific cells and try to ask questions, how is their phenotype being affected ex vivo or to a given stimulation? So yes, we do not take into account, or we cannot take into account the effects of other neighboring cells. And yes, that actually does change the cell phenotype. In our case, however, when we did the single cell sorting, we did it immediately into a lysis buffer. So we took a given tissue, we dissociated the cells, but all given cells were still inside the tube, mixed together or condensed together. And then when we sorted the cells, we sorted them immediately into a lysis buffer. Regarding, future studies or other things that we are doing, yes, these things need to be taken into account when you isolate cells. So you need to take into account that their phenotype might change. So the way that we try to tackle this approach, so for instance, when we do a more pointed analysis on specific cell population, we isolate the cells, we perform the single cell RNA sequencing, but we also do a bulk RNA, or not a bulk, we take the entire tissue and perform this type of analysis as well, and then we can identify those specific population within the given tissue, and we compare the two populations, the ones that are in its natural state in the whole tissue, how those cells, let's say if we talk about specific T-cells, and how those cells phenotype look when we isolated them. And I have to say that in most cases, when we do this thing relatively rapidly, we don't see any differences. I hope that answered the question.
Thank you. Okay, do you think that in vivo might appear fusion between neoplastic cell and normal cell, or even neoplastic cell immune cell, similar to the hybridome process?
I'm taking a moment to review the question one more time. So I want to make sure I understand the question. So if I understand, so do we think that in vivo might represent some sort of interaction between the cancer cell and the non-transformed cancer cell? Or the cancer cell, normal cell versus cancer cell immune cell, similar to a hybridoma process? I'm not sure I fully understand the question, so I don't want to speculate too much. Yeah, I'm going to have to say that I appreciate the question, but I might need it revised from our inquirer. I'm not sure I fully understand the question and how that relates to a hybridoma process. I apologize.
Sure, and we can always follow up with that question after the broadcast, so we'll follow up with you via email. All right, next question. How are the cells from the patient tumor processed to prepare cell suspension them for cell sorting?
Can you please repeat the question?
Sure. How are the cells from the patient tumor processed to prepare cell suspension for cell sorting?
Okay, so that's actually a very good and important question that can actually affect the downstream analysis and quality of your cells. So we're actually applied or modified a little bit the Milteny tumor dissociation kit. So we are using those reagents inside the kit, which are very good. They're very gentle on the cells. They do not remove cell surface markers that other conventional digestion reagents do, which is important if you want to incorporate antibody labeling to your single cell. One thing that we did do and adjusted to our method, and we tested it on several times, and we compared the results that we had, was one, to reduce the time of dissociation. Instead of doing the dissociation for 45 minutes, we were doing the dissociation for 15 to 20 minutes. We also changed the way that we do the dissociation. So if you go through the Milteny tumor dissociation kit protocol, you'll see that they're using the C tubes. The C tubes are tubes that enable both enzymatic and mechanical digestion at the same time. But the problem is that sometimes when you get really small biopsies or needle core biopsies, which are very tiny, if I'll put the biopsy inside, I'll probably lose most of my cells there. So what we adopted, we're actually using Eppendorf tubes that we cut the tissue inside the tube. We add the enzyme mixture at the same ratios, and then we do the dissociation using a thermomixer at 37 degrees, again for 15 to 20 minutes. And I think there's a really importance for how long your dissociation is being done, because we do want to exclude as many artifacts that are being generated by the dissociation itself. So we minimize the time from 45 minutes to 15 to 20 minutes. And in the paper that we published, there's actually a very detailed explanation for the time, duration, and concentration on all the relevant catalog numbers that can annotate for this specific procedure.
Great, thank you. How do you take care of the extracted T-cells for storage and shipment?
So for that, we're using actually a conventional freezing media, which is usually 90% serum, heat-inactivated serum, and 10% DMSO. We tend not to freeze a low number of cells, as these are, when we thaw the cells, so the problem is that you lose number. You can sometimes lose somewhere between 30 to 50% of your cells when you thaw them, so don't freeze a very few cells. I recommend freezing amounts ranging from 500,000 to 2 to 3 million per tube in 1 ml of freezing media. In terms for shipments, so when we do this type of shipments between labs or collaborators, we usually ship those on dry ice. And the cells themselves, they're stored at the beginning at -80, but then for longer storage periods, we store the cells in liquid nitrogen.
Are there any tips you can share to improve SMART-Seq2 results on single cells?
So actually I do, and getting to exactly that, that the detailed reagents and procedure, these are all publicly available in the paper that we published. There's actually a supplementary table. The last supplementary table in our paper actually has a very detailed on what reagents, we used and exactly the temperature for each step reaction that we did. And one thing that I noticed recently that people, when they do SmartSeq2, and that's an issue that I've encountered with collaborators, sometimes they take the protocol from a given paper and they use a reverse transcribed enzyme, which is different from the one that was being used or was not adjusted to the temperatures. So the most common, that's the most common error that we do see. So I highly recommend, if you are changing your RT enzyme, just make sure that you're using the temperature that this enzyme was can work efficiently. For instance, the Maxima enzyme, which that's the enzyme that we are using, that's a very cost-effective one, works best at 50 degrees. And when we talked to a lot of collaborators that had those issues, we actually noticed that they've been using the old protocols and worked at 42 degrees. Another two tips that I can give just to increase the quality of the cells, and this is something that we've been noticing since we had a lot of experience with that, is first work as fast as you can with the first steps. Minimize the time for tissue dissociation. Make sure that your gating strategy is correct, excluding dead cells and cells that are, that you are sorting the cells that you desire. And I think the example that I gave was with the CD235A that exclude erythrocyte progenitors that sometimes people mistake for being the tumor cells. So the two critical steps are actually the tissue dissociation, the quality of the sorting, and from there it's just going by the protocol for RNA extraction, cleanups, cDNA amplification, and so forth. But those two steps we've found to be the most crucial ones.
Great, thank you. It looks like we have time for a few more. What is the best approach to increase the efficiency of plate sorting when using patient sample?
Okay, so especially with the only cell sorter, so this is where we had all our experience. So when you go to the default plate sorting, so this actually gives you an error of 20%. And what it means that if we look at the droplet that contains one cell, for single cell sorting, if the cell is in the center of this droplet, and when we use the default settings, it means that it can move from the center 20% up, 20% down, but not more than that. So if the machine identified the cell not to be exactly at the center of the drop, or with a deviation of 20%, it will abort and won't sort the sample. One thing that we've been doing, we increased a little bit the threshold. We did this like as a custom sorting to 60%. So this range actually enables more flexibility to increase the yield of how many cells are actually being sorted into the plate, and still without losing the quality, meaning that there's still one cell in a droplet. The cells are relatively small to the droplet size. So even within this 60% deviation up and down, we can still capture the right cells and increase the yield of the cell sorting. And again, another thing, as I mentioned in the previous question, is, again, work fast on the dissociation. Start the sorting as once you're done. Minimize the times in between. And that's it, pretty much.
Great, thank you. It looks like we have time for one last question. How should we balance the performance parameters when conducting cell sorting experiments, such as yield, efficiency, purity?
So that's actually a good question, and it relates to the question that I answered before. So there's the custom setting, as I mentioned, so you can actually change that and increase the distribution of a cell in a given droplet, to give it more flexibility to capture more. That actually increases the yield without actually affecting the purity of the populations. If there is a population that's very rare and your sample is super necrotic, so I would highly recommend using the super purity mode. that make sure that, droplets above and below the sorted cell are empty. There's nothing there. So that actually very increases the purity. But then again, it comes on the price of the yield. So sometimes, if you don't want to lose your precious sample, especially if the cell yield is very low, it's a give and take, right? So how much am I putting on the purity and how much am I putting on the yield? So sometimes we need to compromise. But that's the great thing about single-cell RNA sequencing, because since we are sorting single cells into individual wells, and eventually we do analyze single cells, we can actually know if we sorted the right or wrong cell. And I hope that answered the question.
Thank you again, Dr. Feldman and Dr. Jenkins, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.